Now, when we look at that question, it says that with reference to the structures in a chloroplast, explain how the energy from light is made available in ATP molecules for the synthesis of organic materials. Now, in a question like this, from what we have done previously, you will understand that it is asking on the various structures of the chloroplast, like in this case, when you look at the chlorophyll molecules that are found in the chloroplast, that's the photosystems. When you look at how they are used in those redox reactions and how the hydrogen ions move from the lumen to the stroma, and how they move through the special protein, and how they generate that energy in the form of ATP as they move from the stroma, that is from the lumen to the stroma. Now, a question like this, you are meant to be very keen on the points that they are referring to. Like here, they are referring to the structures in a chloroplast. Then they are going ahead and talking about how the energy is made available in form of ATP molecules for the synthesis of organic materials. Now, the first case there, you can see we are saying that the answer to that is that the chlorophyll molecules or photosystems are in the thylakoid membranes and they absorb energy. So it is the chlorophyll molecules that absorb the energy and they are found in the thylakoid membrane. So in that case, you are referring now to the structure itself. Then the electrons in these chlorophyll molecules are excited, emitted, and passed on to electron acceptors or carriers, where they pass from one electron carrier to another in a redox reaction in the thylakoid membranes. Now, what are we referring here? Here we are talking about the electron transport chain. That when the chlorophyll molecules are excited, then they get emitted because of the energy that they possess. Then from there, they are passed through the electron acceptors or carriers. That's as they pass from one electron acceptor to another, you realize that they will lose the energy. Now, when an electron acceptor picks up an electron, we say that it becomes reduced. And when it loses the electron, it becomes oxidized. And in that case, we say it's a redox reaction. Now, this one occurs in the thylakoid membrane. So you should be very keen on talking of the electron transport chain, how those chlorophyll molecules release the electrons, how they get picked up by the electron acceptors, and how they pass through the electron transport chain from one carrier to another, releasing energy, which is used to synthesize the ATP. Now, there is another thing that you need to note, that as those electrons move from one carrier to another in the thylakoid membrane, they release the energy. That energy that is released is used to activate the proton pumps in the membrane. This is what I said in our last session, that when the electrons get emitted or released from the thylakoid membrane, that is the chlorophyll molecules, they get excited, that is after gaining the energy. So they start losing that energy as they move from one carrier to another. So as they emit that energy, you realize they are going to activate the proton pumps. And the proton pumps in this case are those ones that are involved in they allow the movement of hydrogen ions to move from the stroma to the lumen. Now, the hydrogen, so it activates the proton pumps in the membrane that move hydrogen ions from the stroma into the thylakoid lumen, which together with hydrogen ions from photolysis of water create what we refer as an hydrogen ion gradient. And this hydrogen ion gradient, we refer it as an electrochemical gradient, which has been mentioned down here. We refer it as an electrochemical gradient. Now, I want you to understand that electrochemical gradient that has been created, it causes the hydrogen ions to move now from the lumen to the stroma. Now, why do we have this electrochemical gradient? It's because of the hydrogen ions from the photolysis of water and the hydrogen ions from the stroma. They enter into the thylakoid lumen, and that concentration of hydrogen ion increases in the lumen. So what will happen next? An electrochemical gradient is created. that electrochemical gradient will cause the substances now to move from, that is the hydrogen ions in this case, that is the protons, to move from where they are highly concentrated to where they are lowly concentrated. And this one will occur through this special protein which we refer as the ATP synthase. So the ATP synthase will act as a channel protein, and at the same time it will act as an enzyme to catalyze that reaction between the ADP and the inorganic phosphate to form ATP. That's why we are saying here, releasing energy that combines ADP and inorganic phosphate to form ATP that is catalyzed by ATP synthase. And this is what we refer as photophosphorylation. Now, that is a sample question that you can find in some of these past papers, and you can find many more. But once you understand the concept about the photosystems and how the electron carriers work in the electron transport chain, and how the energy is synthesized due to the movement of those hydrogen ions, then it will be very easy to answer many more questions. Now, I want us to look at the flow of electrons and ATP formation. Now, one thing I want you to understand, that ATP formation that occurs in the thyroid membrane in the electron transport chain, it occurs because of the movement of electrons. And that's why I'm saying in the first statement here, that ATP formation in the thyroid membrane, that is photophosphorylation, is due to the flow of electrons. Now, ATP can be formed in two ways, either through cyclic photophosphorylation, or alternatively through non-cyclic photophosphorylation. Now, when it occurs through cyclic photophosphorylation, in this case you will note that the electrons are flowing in a cyclic manner, in a circle. So they keep moving around, they are excited, released, picked up by electron acceptors, they lose the energy, and they come back again to the chlorophyll molecules that had lost them. But when we talk about the non-cyclic manner of flow of electrons, in this case, once the electrons have been released by the chlorophyll molecules that had lost them, once they are released actually by the chlorophyll molecules, they will not come back. Instead, those electrons are replaced actually by those that are released by the splitting of water molecules. And that is now what we refer as the non-cyclic photophosphorylation. The electrons, once released by the chlorophyll molecules, they do not come back to the back to the chlorophyll molecules that are lost to them. Instead, they are replaced by electrons from the splitting of water molecules. Now, in the cyclic and non-cyclic photophosphorylation, we have two stages of the light-dependent stage. And this one we have the light-dependent stage and the light-independent stage. Now, first I want to look at the non-cyclic photophosphorylation, and then we look at the cyclic photophosphorylation. Now, when we talk about the non-cyclic photophosphorylation, first let me remind you that this photophosphorylation is the synthesis of ATP with the use of sunlight energy, and whereby the ADP and the inorganic phosphate are joined together, and this occurs specifically in the thyroid membranes. Now, when we talk of photophosphorylation, which is non-cyclic, it means that the electrons, once they leave that chlorophyll molecule, as I said in our previous slide, that the electrons, once they leave a chlorophyll molecule, they do not flow back to reduce the same molecule, but other electrons reduce it. Where do those other electrons come from? I've said they come from the photolysis of water molecules. And in photolysis of water molecules, light energy is used to split the water molecules to form hydrogen ions, electrons, and oxygen gas. Now, those electrons are the ones that are used to replace those ones that have been lost by chlorophyll molecules, because those ones, they do not come back. Now, the photosystem's P700 absorbs light energy, and we said that the P700 is actually on a higher ground state, and we said that the electrons here are easily excited. And you remember we talked about the primary pigments and the secondary pigments. So when you look at the P700 of photosystem 1, we said that the P700 is actually photosystem 1. It absorbs the light energy, and once it absorbs the light energy, the electrons now become excited, they get released or emitted, and they are passed to the primary electron acceptors. Now, the primary electron acceptors, in this case, they are the ones that pass them now to other electron carriers, and in the process, as that energy, that those electrons are passed from one electron carrier to another, energy is lost. And that energy that is lost is used actually to drive the synthesis of ATP. Now, the electron combines with a hydrogen ion or a proton from the photolysis of water to form hydrogen atom that reduces NADP. Now, I'll take you to one of the slides that we looked at in our previous session. This one here, where we were saying that the electrons from photosystem 1 now, in this case, they combine with hydrogen ions from water. That these electrons now, they combine with hydrogen ions from water, and these electrons are from the P700. Then they form hydrogen atoms which link with NADP to form reduced NADP, and that reduced NADP is the one that proceeds to the Calvin cycle. Now, that is what happens in the non-cyclic photophosphorylation. Now, I want us to look at our next slide. This one talks about the P680 of photosystem 2. Now this one, the photosystem 2 usually absorbs light energy. Its electrons equally get excited, they get emitted, and they are passed on to primary electron acceptors, just like what appears in the P700. Once the primary electron acceptors pick the electrons, they are passed to other electron carriers, and then they are passed from one electron carrier to another, and in the process energy is released, and that energy, as I said earlier, is the one that is used actually in driving the synthesis of ATP. Now, what happens is that the photosystem 2 is the one that is going to receive the electron that was initially lost, reducing the P700 of photosystem 1. How does this one take place? Let me take you to another series of reactions here so that you see what I mean on my next slide. Now, we have said that the electrons from photosystem 1, this one that is in this diagram here, the electrons from this photosystem 1, they are lost when actually the photosystem 1, that's these pigments, they gain energy. Once they gain that energy from the light, the electrons get excited, they get emitted, then they are passed to the primary acceptors. Now when they get past the primary acceptors, they are passed from one carrier to another, and in the process they drive the synthesis of ATP. But in the process, these electrons that have been excited from photosystem 1, they are picked by the hydrogen ions here from water, and once they are picked by the hydrogen ions from water, they form reduced NADP, and that's why we are saying that this NADP is an electron acceptor. Now what happens next is that this reduced NADP that has been formed, it is taken to the Calvin cycle, that is the light-independent reactions. Now, this is for the P700 photosystem 1. Now, for photosystem 2, this one here, the electrons released by, when light energy falls on this photosystem 2 pigments, they are going to gain energy, they get excited, they get emitted, and pass to the electron acceptors, which will pass the primary electron acceptors. Now, from the primary electron acceptors, they are passed to other electron carriers along this electron transport chain in our previous sessions, whereby we said it's like a staircase, and that energy is lost as those electrons move from higher energy states to lower energy states. And in the process, the energy that is lost, it is used in driving the synthesis of this ATP from ADP and inorganic phosphate. Now, that electron that was lost from photosystem 1, you see, once it passes through the electron transport chain, it will be used to replace the electrons that were lost from photosystem 1. Now, where are we going to get the electrons to replace these ones for photosystem 2 here? The electrons will be released when, for example, the water molecules are going to undergo photolysis, whereby the water molecules will undergo splitting through the process of photolysis to form hydrogen ions and hydroxyl ions, and in this case, energy will be lost. Not energy, sorry, electrons will be lost in the process, and oxygen will be lost to the atmosphere. These electrons that have been lost from here, they will be picked by the photosystem 2 to replace those ones that actually had been lost in photosystem 2 here, and were used to replace those ones that were lost in photosystem 1. Now, these electrons are obtained, you can see from here, from the splitting of water molecules in the process of photolysis. Oxygen is actually released to the atmosphere, and in some cases water, if it is hydrogen ions and hydroxyl ions formed, or directly you can say water splits to form oxygen, electrons, and oxygen, hydrogen ions, and electrons. This oxygen is used in the process of respiration. Now, these hydrogen ions at this point, they will move actually and be picked up by the electron acceptor NADP, and this one occurs either when they gain hydrogen ions, they gain electrons from photosystem 1, or alternatively, even without gaining those electrons from photosystem 1, they can bind direct to form NADPH positive, meaning that they did not gain the electrons. Now, I want us to look on our previous slide still. So if you look at this, we are saying that the P700 of photosystem 1, this one absorbs the light energy. Its electrons become excited, emitted, and passed on to primary pigments, that's the primary electron acceptor, which passes it to other electron carriers. The electron combines with a hydrogen ion or a proton from photolysis of water to form hydrogen atoms that reduces NADP. Now you can see exactly what we said is that the electron that has been obtained from photosystem 1, it will combine with an hydrogen ion or a proton that was obtained from the photolysis of water. Actually, the ones now we were referring here, that was obtained, the hydrogen that was obtained from the photolysis of water. So this electron here will combine with it, and then it will form an hydrogen atom that reduces NADP. And that's why now we end up finding reduced NADP at this section here, after this electron combines with this hydrogen from water to form reduced NADP, and this one is taken to the light-independent reactions. Now, if you look at our next slide here, which I was talking about, the P680, to explain further on that diagram, you can see the P680 of photosystem 2, this one absorbs light energy, and its electron becomes excited. emitted and passed on to primary electron acceptor that passes it to other electron carriers, then transferring it through the electron transport chain, driving the synthesis of ATP. The photosystem 1 then receives the electron that was initially lost, reducing P700 of photosystem 1. That is the same thing that I was talking about here. That if you look at the first thing, or about the getting excited is something that you're already aware of, that once those electrons get energy, they get excited, emitted, and passed on to primary electron acceptors, and this one passes it to the electron carriers. Now, once it passes now through the electron transport chain, it will lose the energy that is used to drive the synthesis of ATP. Now, what I want you to note here is that the photosystem 1, it is the one that is going to receive that electron that was initially lost from the P700. So the photosystem 1, once those electrons have been lost, what is going to happen is that the photosystem 1 will now receive that electron here and to replace this one that was lost when light fell on this photosystem 1. That's the P700. Now, if you look at the next part here, you see we are talking about that at the same time the electrons in photosystem 1 are excited and collected by the electron acceptors. So you see in photosystem 1, you remember the electrons were lost from photosystem 2 and also they are being lost from photosystem 1. But you see these events must occur at the same time. Now, if they occur at the same time, it means that we must have the electrons being lost from photosystem 1, at the same time being lost from photosystem 2, and at the same time the water molecules undergoing photolysis to produce electrons to replace those ones in photosystem 2. Now, that's why we are saying that at the same time as the electrons, at the same time the electrons in photosystem 1 are excited and collected by the electron acceptors. Now, which electron acceptor is this? Is actually nicotinamide adenine dinucleotide, NADP. It picks up that. But before it picks those electrons, they must pass through what we pass the electron transport chain. So they are picked up by the primary pigments, then picked up by other that is the primary acceptors, then they are picked up by other electron acceptors or carriers, and then they are passed on to the NADP. But before they are picked up again by NADP, remember, they need to join by the hydrogen ions to form hydrogen atoms. Now, there is a part of photolysis of water. This thing is like a cycle or a circle whereby we cannot say exactly this where the reaction starts from, because these things occur simultaneously, that is at the same time. Now, the photolysis of water, we are saying here, it takes place in the thylakoid lumen, producing hydrogen ions and hydroxide ions. Remember, it is the hydroxide ions that will undergo oxidation so that they can form the electrons, which are actually transferred to photosystem 2, which is now unstable, as I said earlier in our diagram actually here, whereby I said that those electrons are used to replace those ones in photosystem 2 that had been lost, so that it can stabilize these chlorophyll molecules. Now, we are saying that once the hydroxide ions lose the 4 electrons, meaning they undergo oxidation to form water and oxygen, the electrons are transferred to the unstable chlorophyll molecule in photosystem 2, restoring its stable state. So it's all about restoring stability, because you realize when an atom sometimes loses electrons, it becomes unstable. But this one now depends on how many electrons it had before from your chemistry. Atoms lose electrons to be stable, and at the same time they can lose electrons to be unstable depending how many electrons they had initially in the outermost energy level. But now here, the electrons are transferred to the unstable chlorophyll molecule in photosystem 2 because it had lost the electrons and it became unstable, so that this one is meant to restore its stability. Now, in this case, you see, we are saying that the electron reduces the P68 of photosystem 2. Now, in this case, it reduces the P68 of photosystem 2. Now, the electron I'm referring to is this one that is being obtained from the splitting of the water molecules. Now, it is used to replace those ones that were lost in photosystem 2, meaning it reduces the P680. Then the hydrogen ion combines with the electron from P700 to form hydrogen atom, which reduces NADP. Then the oxygen is released to the atmosphere through the stomata as a byproduct or used for respiration by the plant. And I want you to note on this that cyclic photophosphorylation and cyclic photophosphorylation, they seem to work together, because actually they realized that there is photosystem 2 later when they realized that there are electrons, there is photolysis of water, and there is electrons that are released from water molecules that are used to replace the electrons in photosystem 2. Now, in the cyclic photophosphorylation, you would realize that this one, there is a flow of electrons in form of a cycle, and this one occurs in photosystem 1, meaning that the electrons from photosystem 1, they do what? Once released, they flow back to replace themselves after they have lost the energy in the electron transport chain. And that's why we are saying that the cyclic photophosphorylation, it involves photosystem 1, and it involves the cyclic flow of electrons from P700 of photosystem 1 and back to the same molecule to reduce it. Now, the product that is formed is ATP, meaning that there is no other product that is going to be formed in this reaction. Now, for example, you can see if the electrons are flowing through the electron transport chain, we are not going to form NADP. So it means that the cyclic photophosphorylation will only form ATP. And that's why we say this photosystem 1 is a bit useless to the process of photosynthesis, because if it does not form reduced NADP, which is required actually in the next phase of photosynthesis, then it is unnecessary. It's a waste of time. Now, the P700 of photosystem 1 absorbs the light energy, then its electron becomes excited, emitted, and passed to primary electron acceptor, which passes it to other electron carriers, forming ATP, and finally it reduces the P700. Now, what we are seeing here is actually the cyclic type of photophosphorylation in this diagram of mine, whereby once the electron is excited from the chlorophyll molecules, the electrons lose the energy. Once they lose, not the electrons, but the chlorophyll molecules lose the electrons gain that energy, they move to higher energy states. As you can see in this diagram, they move to higher energy states. Then from there, they actually move through the electron transport chain, and through the electron transport chain, this is where they lose the energy that is used to drive the synthesis of ATP from ADP and inorganic phosphate. And that electron, once it has lost that energy, it again comes back to the chlorophyll molecule that had lost it. Now, when you come to the next diagram here, I'm showing how the cyclic photophosphorylation occurs. And this one is almost the same thing that we saw there, but it's only a change of the diagram. that when the light falls on the chlorophyll molecules, they get excited to higher energy levels. From here, once they get excited to higher energy levels, they start losing the energy as they pass from one electron carrier to another through the electron transport chain along this place here. Once they lose that energy, it is used now to drive the synthesis of ATP from ADP and inorganic phosphate. And that is now when we talk of the electron carriers in this case. These electron carriers, they again come back to help in picking up those electrons at this point. So the chlorophyll molecules here, you realize that they again regain the electrons that had been lost earlier on, and this is the cyclic photophosphorylation. If you look at the non-cyclic photophosphorylation here, you realize that the electrons from the chlorophyll, once they get excited by the energy, the light energy that falls on them, they get excited to high energy levels, and then they are passed from one electron carrier to another along the electron transport chain, synthesizing the energy, actually losing the energy and synthesizing ATP. And you realize that when they move to lower energy states, they do not go back to the chlorophyll molecules that are synthesized to them. So they are not flowing in a circle. Instead, they are picked up by the hydrogen ions that were obtained from the photolysis of water molecules. And those hydrogen ions are actually, when they gain those electrons, they become reduced. And those hydrogen atoms now that have been formed, they join with NADP, which is oxidized, to form NADPH. This NADPH is the one that is taken to the light independent stage, so that now it can be used in carbon dioxide fixation. Now, these water molecules, remember, they are being split through the process of photolysis to release hydrogen ions that pick electrons from this photosystem 2, and the oxygen is released to the atmosphere for the process of respiration. These electrons that are released from the photolysis of water molecules here, they are used to replace those ones that were lost actually by the chlorophyll molecules. And that's now what we pass the non-cyclic photophosphorylation. Now, the next slide here, we are talking about the role of the products of the light-dependent stage. And this one, I want you to, that's the role of the products of the light-dependent stage. I want you to understand that two of those products are taken to the light-independent stage. The one that does not require, that does not necessarily require light for it to take place, that occurs in the stroma. But the other product, which is oxygen, is actually released and used in the process actually of respiration. Now let's look at the first case here, where we are talking about the NADP. Now, reduced NADP can also be referred as NADPH. And this one is actually formed when hydrogen atoms combine, that's hydrogen ions combine with electrons to form the hydrogen atoms. Then those hydrogen atoms are the ones that combine with NADP to form NADPH. Now, I want us to look at this, that the ATP that is formed and the reduced NADP from the light-dependent reaction, they are used in the light-independent reaction in the conversion of glycerate 3-phosphate, which we refer as GP in short, into GALP, that is which is glycerate 3-phosphate. And this GALP is the one actually you realize later that is used in the formation of the exotic sugars like glucose. So that those exotic sugars are now used actually in the formation of biological macromolecules like let's say carbohydrates, lipids, proteins. You'll see how that one occurs in our next sessions. Now, oxygen is usually released to the atmosphere and then it is used as a byproduct. And I think on that section we are done on the role of those products. Now in our next session, next slide, sorry, we are comparing on the cyclic and non-cyclic photophosphorylation. Now, if you look at these two cases, the cyclic and non-cyclic, first I want you to understand that the non-cyclic photophosphorylation, it involves both photosystem 1 and photosystem 2. But when you look at the cyclic photophosphorylation, this one involves photosystem 1. Remember, photosystem 1 is the P700, but the non-cyclic photophosphorylation is the P680. Now from there, you can actually know when we talk about non-cyclic, it means the electrons do not come back to replace those ones that have been lost. So the electron that was lost does not replace itself again in that chlorophyll molecule. Instead, it proceeds to the next phase whereby it replaces that. It's actually picked up by the hydrogen atoms. Now, when you look at this cyclic photophosphorylation, the electrons come back and they replace themselves. And that means that they are not involved in the formation of reduced NADP, and that's why it is a useless stage in photosynthesis. Now, let's see the differences. This non-cyclic photophosphorylation requires light as the same as the cyclic photophosphorylation here. Both of them, they form ATP. Both of them, they involve the flow of electrons. Both of them, they involve photosystem 1. But now the difference comes in photosystem 2, it only occurs in non-cyclic photophosphorylation, while cyclic photophosphorylation involves only photosystem 1. Now, non-cyclic photophosphorylation, it involves the photolysis of water molecules, so that those electrons from the photolysis of water molecules are used to replace those ones that have been lost in the chlorophyll molecules. But when you look at the cyclic photophosphorylation, there is no photolysis of water molecules. So it means that the as we said earlier on, the electrons move in form of a cycle. That is a cycle. That means that there is no need of replacing them using an external source. Now, the products of non-cyclic photophosphorylation is oxygen, ATP, and reduced NADP. But when you look at cyclic, it only involves the production of ATP, and that's why I said that this one is a useless stage in photosynthesis. Now, when you look at the non-cyclic photophosphorylation, electrons, they do not go back to the reaction center, but so they actually used in the formation of reduced NADP. But you realize in the cyclic photophosphorylation, the electrons flow in a cyclic manner and they go back to the reaction center. You remember we talked about the light harvesting center and the reaction center. So they go back, these ones actually, to the reaction center. And that's why, because of this non-cyclic photophosphorylation, that it produces so many products, that is especially those that are required in the light independent stage. That's why we say it's a useful stage for photosynthesis, because it produces ATP and reduced NADP. And when you come to the cyclic photophosphorylation, this one you realize that it does what? It is a useless process in photosynthesis because it only produces ATP. And that brings us to the light dependent stage. In our next class, we will actually look at the light dependent stage, and that we stop there for this group.